I'll wait for this to shift over, and I want to say thanks everybody for joining today. I hope I do make it worth your while and your investment of your valuable time as well. And thanks to Krishna. I was granted the opportunity to visit University of Illinois Chicago in his lab last year and give essentially a similar talk. And I was thrilled to do so, and this is a great opportunity to share some of that information more broadly. So what I'm going to talk about today, and I've put this together as a presentation that's kind of a sampler of a lot of work we've done. So this is well over a decade, the better part of 20 years represented here from the first information we've done through the end. So none of it is at great depth or everything fully explained, but I give the references in there, and I can share those out through Krishna as well. And I'd also give a shout out. We're hosting the International Fighter Remediation Conference in India this fall, and I'll send some of that information through Krishna, where we've got a lot of this information, research, and much more. It's great that it's the first time it's been in Southern Asia, so we're thrilled to do that. So I'm going to represent two ideas today. Everything that's presented here really has connection of two topics, and that's human health and plants. So how do plants and human health kind of come together? And right now, I want to draw your attention to one thing that I hope is kind of a focus now and going forward, is the plants have the unique ability to be a great energy transfer that crosses over between the atmosphere and the subsurface. And very few things actually do that in nature, but plants have that ability, and they've got intimate contact with both. So that's the key focus we're going to look at, and everything from that kind of builds on how do we take advantage of that by understanding the science of it, and then looking forward at how do we engineer opportunities for us to take advantage of that. So the first topic is plants. And just to give a little bit of a background here, some may be plant physiologists or horticulture, but I'm going to go forward with that. And what you see in there is a very small seed in the palm of my hand. And that is a redwood seed or a sequoia seed. So that little tiny seed can become the biggest and the oldest organism on our planet. And truly, that's pretty amazing. And to think about that seed never goes anywhere else. It harvests everything it needs to do that to become this. So this is actually a tree. So this tree is the General Sherman tree. It's in California, and it's estimated to be over 2,400 years old and over 2,000 tons now. I should update that. It's actually grown. So it's a substantial organism. And again, it grew there without moving at all. And plants are also amazing at how they essentially populate almost the entire planet. So these are some unique ones. The one on the left is called the resurrection fern, and that can grow in a desert and be dormant for years at a time, get one rainfall, go through a full vegetative state, and then go dormant again with the seeds waiting for the next rainfall, which again may be years away. And the other two on the right are actually two vascular plants that grow on Antarctica, and these can be covered for 9 months of the year, entirely frozen, and then come back to life and go through an entire vegetative process and be ready for the next year. So the key thing to take home here is that plants occupy anywhere on the planet almost, and the same places we do. And again, they're tied between that subsurface. And this is a little video that I just pulled from the web. I give it acknowledgement later. But these plants, think about what they're doing here is they're sensing all the things you can see on the left. They're sensing light, temperature, gravity, nutrients, water, and in many cases, each other. So as they're growing, they know where gravity is, and the roots go down towards that. They're sensing oxygen the whole time and the availability of water, and they'll change their structure based on what the soil type is and what the water availability in the soil type is. As we see the cotyledons coming up through the surface, they're sensing sunlight and temperature and growing, modifying their growth based on those feedbacks. So the key point there is that the plants are active. So I think when we look at them, they're generally not moving or doing anything else, but they're very active, and they're searching out things in the environment all the time. Now to bring in the second topic, and that's the human health. And our environmental disease burden comes from all over. It's a variety of exposures that come through different media. So we've got air, water, contact with soil or surfaces or each other, and soil, through the food. So we also get soil particles in the air. But all of these media then carry to us some environmental burden, be it pesticides, herbicides, natural organic matter, radioactivity, viruses, bacteria, compounds that are endocrine disruptors, irritants like ozone or particulate matter, all come through these pathways. So if we look at the built world and where we're at and what our exposures are, they can come from contaminated sites, and these environmental pollutants then can get through our groundwater and come up through our drinking water or into our home, or they can be particulate matter that's blown around and contacting us. So again, when we talk about some of those environmental burdens being anthropogenic or man-made in origin, they can get to us in a variety of ways. So when I'm talking about this, I'll draw your attention to the person in the home, the person outside in these areas, and the tree. So the tree occupies a similar part of the biosphere that we do, again, crossing over to that shallow subsurface where pollutants can get to us via different mechanisms, but also the atmosphere around us that we're exposed to as well. So that brings us to that idea. So if we've got plants that occupy the same part of the planet as we do, and there's pollutants in that environment, which we know there are, we're exposed to the same pollutants as that tree. And as that tree is place bound, and as I mentioned, very active at harvesting everything it needs from the subsurface or from the atmosphere, it's actively, in many cases, harvesting some of the pollutant molecules or atoms that are in the surroundings. So if we look at how those compounds move around with us, and we base some of the information on chemical properties such as vapor pressure or solubility, we can see a variety of exposures for that plant. Well, there we go. And again, as dry air comes through, it becomes moisturized by the plant. So it's actively pumping some of the groundwater up to the atmosphere. And that pulls not just the groundwater, but also those pollutants above ground. And that's where we step in. So now those compounds that were in the subsurface, and for some reason my pointer's not going very well, that were in the subsurface are now in the superficial aquifer, or excuse me, above ground, where we can get at them in this way. And that's where we come in. So we're looking at different methods where we can come to that tree and say, how can we get at some of the compound information that is in that tree as far as what the groundwater pollutants were. And if we can relate those groundwater pollutants very rapidly to the above ground surface, we can monitor things pretty quickly and efficiently. Sorry for a little bit of a catch up here. My mouse died and I can't forward very well. Now we should be back in there. So what we do, you can see we're on a polluted site here. This is actually in Ontario, the Burken site in Canada. And you can see that there's a lot of groundwater monitoring going on. So this is a known site, but we sampled the plants there with an increment bore, and that just pulls a small section of the tree out. You can see in the picture there. This is all we leave behind is a small hole in the tree that does cause some damage, but generally not impacting the tree. And we bring it into a variety of different then analytic techniques. So here we're showing a VOC vial, and we're collecting volatile organic compounds at this site. So then we can run analysis on this, and literally within a day of being on a site, sometimes we can have the data ready to plot out. And this would be a great place for some questions, but we'll just keep moving a little bit. And I'll show you one of the case studies. And this was actually one of our first ones and one of our more interesting ones, and we've done a lot. I won't go through all those case studies. We'll get into some new science. But in New Haven, Missouri, this is on the Mississippi River, so it's right in central Missouri. The two city water wells that were available showed up with chlorinated solvents, mostly perchloroethylene at the site, 1987. So the wells were closed and new wells were drilled to supply water for the city, but the source of the contamination was unknown. So now we're going to zoom in just to this area right by the riverfront, and this is where we come into it. So this is tree coring that was done. So this is an area that's populated. These are homes. This is an area where the some of the compounds that were used had been used in a small industry. And we cored all of these trees in basically a day. And you can see certainly just kind of showing a heat map where the contamination was and was not in the trees. So that directed then the groundwater drilling and analysis that was done, some through the slab of the building, and sure enough, there was a source area there. But you can see it's very efficient. So based on where the trees were, you can see that tree core plume where we've got a circle of all of our hits. That's exactly where the groundwater was. So there was essentially no wasted analysis here at directing it. And again, we knew where it was from the trees. We just couldn't quantify exactly how much. And this just shows what that site looks like. So these are people's homes. These are the trees that are in their yard. And from that, there's the groundwater plume and the heart of it based on this. And this is a flood protection levee. So this is a large river, the Missouri River, that does flood this area time to time. So to back out to a city view again, that was not the source of all the pollutants though. So I'll back up and say this was very shallow. So it's an alluvial area, sediment from the river floods, floodplain, and the contamination did not go deep. So when they did drill deeper, it just was not at depth. Yet those city wells, again, were contaminated. So these show tree cores from across the community, and the actual originator of some of the pollutant had actually moved a larger operation out here, and they had some pollution in that area. But that actually flowed to the south. And lo and behold, at the high point of the city, we started getting hits again. So these were very high concentrations. And you can see here the student that did that, who's now a professor at California Fullerton. And here's then after they did the groundwater analysis that showed what was going on. So up in this, again, the high point of the city, one of the workers who had been at that industry had actually been taking chlorinated solvents for his own use and then disposing of them just in a superficial dump. And that went deep, and that was the source of pollutant across the city. So this is at depth, again, with deep groundwater wells and monitoring wells put in. And you can see that that is definitely the origin of the contamination. These are the city wells up here that were contaminated. And I would venture to bet that this never would have been found with traditional methods, because again, there's no reason to think that in a residential area you'd have industrial pollutants being dumped, but they were. And you'd never, think of going into people's property with no, reason for suspecting that. And again, it was very efficient. Once we knew where the contamination was due to the tree coring, we could direct the analysis. So I could show many sites. We've done this over 110 sites. Many times we get contracted from people to just do the analysis, and they'll collect the tree cores. It's been over 16 states and 8 countries. And there's guidance documents by the United States and USGS and US EPA and in the European Union called Bio-tools. So this does have documentation and guidance on it. It is not an EPA approved method, but it is an EPA recommended method for doing screening of sites. And so we just had some new contracts for some new sites this summer. We've done more than 15 different compounds, including a patented method where we can do direct analysis in the tree and actually put sampling ports in the tree. And we've done mostly chlorinated solvents and volatiles, but also 1,4-dioxane, a series of explosives, and perchlorate. And with that, I'd just like to shift gears and say we definitely can, if the trees have good contact with the groundwater or the volatile plume, we can definitely pick up many, many compounds in the site. But what else can trees detect for us? So once we understand that it's a good linkage to that subsurface, we can look at different things. So this was a little bit different work we did. And again, this kind of explains that idea of if pollutants are moving into the tree, we can understand where they're coming from. But we also know that vapor intrusion is an issue, and very similar to the trees, the homes actually have a gradient on the vapor phase into the home. Most homes are at a slightly negative pressure, so the pollutants in the subsurface that get pulled into a basement through vapor intrusion can also be pulled into neighboring vegetation. So we wanted to venture out and say, can trees be utilized as a specific indicator of vapor intrusion? So here's just a little bit of a model at different ways to do subsurface investigations, and particularly for volatile compounds and vapors. So we have our traditional groundwater wells, very expensive to put in for a single monitoring point, and also rather invasive to put in as far as the property owner goes. We also have subsurface gas sampling, which is a very small volume. So that is looking at taking out of the ground a couple liters of air, of vapor, and monitoring that. But again, it's very small. And if we look at the groundwater well, it's not really vapor in this case, it's the groundwater. So we don't understand how well that might be tied to the vapor and the porosity or permeability of the subsurface. Then we can look at the home, and we do have sub-slab sampling, where very similar SUMA canisters or SPME fibers can be placed under the building. Again, we're sampling a very small volume. Or we can do the indoor air. Now that has the problem of the compounds in there may be coming from the subsurface or maybe pulled in another product. So in many cases, when we look at for a chlorinated solvent or a petroleum compound, it can be brought in through other avenues, not just the subsurface. And then if we look right next to the building, we've got the tree that is actually sampling a very similar volume to the entire basement of the building at the same horizon. So the plants generally don't put their roots into the groundwater. They put it down to the groundwater table. And very similar, we generally don't put our homes below the groundwater table. We put it down to it or just the standard depth. So that environment that we're really concerned about and the volume of the tree are very similar and have similar contact to the subsurface. And with that, we can be in and out of a property in minutes with very little impact to the environment. So if it is a clean site, we're not invading their property for just coming up with background. So we also want to look at soil vapor volume and sampling. So when we look at a building and the sampling volume of a tree, we had the question of, are they similar and can they be directional, so to say, within that? So we know trees have roots that extend in all directions. Can we look at a single tree and pick that up? So this is some work largely done at Vienna, Missouri, where we had a contaminated site with very few homes nearby, but actually we knew the contamination distribution fairly well. We were contracted to fully do a delineation on the contaminants, so we were able to also look at the vapor sampling. This is primarily the work of Jordan Wilson, who now is with USGS, and was working with them through part of this time also. So a couple different things we want to look at is, can we do a good vapor profiling of a site, and how well is it related to the groundwater, or how well is it related to the vapor? So this is again Vienna, Missouri, and it's a super fun site in the US. The primary source of the contamination was a small industrial facility, and we knew that was the case. So we had some sampling wells going in, but we invested heavily, mostly of our time and effort, to do other sampling. So we did over 900 soil samples with GeoProbe to go down to moderate depth, and over 450 trees were sampled across the site as well. And you can see this mapping, and the triangles are the trees that were sampled, and the circles are the groundwater. And what we detected, again, without any initial groundwater sampling, just with the trees, was that we did have that known area of initial contamination where the buildings were, but we also had two other sites where this is actually a storm drain from this area, releases down gradient or down the hillside. So it's contamination there. And then they had taken the spent solvents and put them on the back of the property and disposed of them, just open disposal. And again, at this point, this was through the 50s, 60s, so there was no environmental regulation based on disposal of these compounds. But you can see it does go off property. The property is shown here, and does go fairly deep at this site as well. And again, this is mostly perchlorinated solvent, or perchloroethylene. And also back up and say, and we also were able to go out quite a ways with this and very cheaply look at, do we have contamination in that subsurface horizon? So it was fairly contained. But when we look at that tree, we wanted to understand, what exactly are we sampling? What's the environmental volume? And if it means, if we detect something or we do not detect something, what does that really mean as far as the area? So the next part of the work looks at, can we have a relationship, now that we have a lot of soil data in this profile, and say, what is the known volume of a tree? And we based it on the diameter of the trunk. So something very easy to measure. And again, it's not a perfect measure to know where the roots are, but it is an indicator. So what I'm presenting here then is this root to trunk diameter ratio. So if we have a known trunk size, how big is the the mass that we're looking at. So to boil this down very quickly, and this is a basically an R-squared model to say where are things detected and what does it mean. And with that adjusted R, if we say if we're 50% confident, it means something. Again, it doesn't absolutely mean it's there. What is the ratio of the trunk? So if we look at the soil samples, and I'll go back here. If we look at the soil samples out from a tree, is it statistically related to the tree concentration? And we said, again, at a greater than 50%, and that's far from perfect, but it is a strong indicator then. And the ratio was 3 centimeters to meter within that. So essentially a 50 centimeter tree did have some statistical relationship to concentrations within 150 meter diameter of that tree. And a 50 centimeter tree is a very large tree in that case. And that gives us an area sampled by that tree of over 17,000 meters squared. So that's a pretty good size sampling area to say if the tree is clean, we have some feeling that the area around it up to 150 meters is clean for that size tree. Or if it's contaminated, there's contamination within this area. But again, we don't know where within that. And we're talking about 150 meter diameter. That can be a substantial difference, especially around an individual home. So the next question was there directionality? So again, if we look at, we've got a very, very large diameter around the tree where there's something, can we look at multiple samples off of a tree and then relate it? So this is the first analysis, and if you can see this little pie shape, that actually says we're sampling different sides of a tree. So in this case, this was a large sample, and we had some contamination in all directions on this one. If we look over here, we had some contamination on one side of the tree, and over here we had some contamination just on one quadrant. So we then advanced that quite a bit and said, what if we took multiple samples? So in some cases, we took as many as 12 samples around a tree. And the data here gets a little bit complicated because you can imagine if I have 12 samples around a tree and up to, in some cases, 20 or 40 samples in the soil around a tree, we wanted to relate where the centroid was. So the next data is going to look at a comparison of the soil concentration. So in this case, you can see the higher concentrations are in this quadrant or directly, quote unquote, north of the tree. In that case, just on the y-axis. And if we look then at what the in-plant concentration is, so the triangles are the tree concentrations, and again, just showing 4 samples around a tree this way, you can see that the tree centroid is over here. And again, if we have as many as 10 or 12, we actually have a little bit better data on the centroid within the tree and relationship. So what I'm going to compare then is that angle of comparison. So are the angles pointing in the same direction for the contamination, or is the tree and the soil not related at all? So again, the idea is if the subsurface is contaminated on one side, is the tree contaminated? And we do know that trees have sectoriality, it's called, where the groundwater on one side of the tree generally tends to stay on one side of the tree. There are some that spiral a little bit, but we do have an indicator there. And we've actually seen that in some areas where there's a lot of road salt applied, and it can kill the side of the tree that's closer to the road. So here's our contamination plot again, and this is the spots of all the soil samples that were taken. And then here's that comparison. So it's easiest to see with this big tree. So the size of the tree is the environmental volume that the tree is expected. So this is one of our larger trees on the site. And then the red arrow is looking at the soil data, and the black arrow is looking at the tree samples. So in this case, you can see that the angles are fairly close, but then if we look at some other trees, the angles are not very tight in agreement. And we looked at then what the gradient was around it. And essentially the next slide gets a little bit more complicated, but it turns out if we have a strong gradient, so in this case, we have high concentrations within the area sampled by the tree and very low concentrations in the other side, the angles tend to agree better. And that would make perfect sense because if there's no gradient, you'd expect no relationship. So really the angle is an indicator of how strong the gradient is in concentration in the subsurface, and it does show up in the tree. And here's that comparison. So again, what we're looking at here is the difference in the angle as related to a difference in the, in the tree, as related to a difference in the subsurface. So if we have a stronger gradient and we come out this direction where that tree 19 is at, so there's a strong gradient, it's a very different concentration on one side of the tree up to the other in the soil, we see a big difference in the tree. If we get down here where there's very little difference in the gradient, we don't see a good angle of comparison. So what this says is if there's not much difference in the tree, there's generally not much difference in the subsurface. And then we just reversed that data and also plotted it. And then this is some actual, and again, there was very little as far as homes go in that case, but we did have the opportunity to have a lot of soil data and a lot of tree data. Now, the next one, we actually go to a contaminated site in York, Nebraska. And if we have more homes, we generally have less trees and we also have less access to get subsurface. But this is showing a map of the subsurface contamination and of the tree core. So you can see out here where the contamination drops off quite a bit, we don't have much in the trees or in the homes. And then we see in this area, it's a little bit more downtown. We do have the hot spots and we do pick the contaminants up in the homes as well, and in the trees. But again, there are fewer trees where it's more populated. And this just shows that data a little bit more condensed. But we did have very good agreement, in if there was contamination in the homes, that the neighboring trees were contaminated. So it's really the first proof that the indoor air was related. And this is some current work in Washington, Missouri, done with USGS on a US EPA site. And you see the color coding for the properties essentially shows the vapor intrusion concentration in the homes, and the dots are essentially the concentration in the trees. So once again, we see a good correlation overall of where the area is. And I will say the variability in the homes is a lot greater than the variability in the trees. And that has to do with how well the homes are connected to the subsurface. Is there ample gas exchange between the subsurface, or are the homes well ventilated? So after this was done, many of the homes had a vapor intrusion mitigation process put in where they actually have an exchange of the gas around the tree, or around the house. And so the concentrations in the trees drop, or in the homes drops off. And my point there is the trees are an indicator of vapor intrusion potential. It doesn't say that there is going to be a contamination in the house because the houses are all very different. So if a house is well ventilated, the contaminants will not build up in the home, regardless of whether or not they're in the subsurface. So the trees indicate the contaminant presence in the subsurface and the potential to be in the homes. And again, this is being used as a screening tool now for USGS and US EPA. All right. So now we're going to switch a little bit, not at do the compounds get in, or what do the compounds indicate, but this switches over to what chemicals should we actually be looking for in the plant and which ones are able to be moved in. Now pause just for a second, say, is there any questions at this point that I could answer quickly before we move on and get to the questions at the end?
So it looks good so far.
Excellent.
Yeah.
All right. And then which contaminants get in and which don't, and what does that mean for that kind of general science? So this was a really nice body of work, and I'd say this is a pretty interesting read. And Matt Lemmer is now Dr. Matt Lemmer at the University of Delaware, and he put this work together initially. So this was our initial kind of screening. So some of this data goes back to my PhD, and essentially there's a lot of scatter in this, no doubt about it. And the general feeling is that the log KOW, or the hydrophobicity of the compound, is related to how efficiently it moves into a plant. So its transpiration stream concentration factor is a ratio of what the exposure concentration is to the in-planta concentration moving in the transpiration stream. So a 1.0 essentially moves that there's no resistance to the compounds moving into the tree through the root structure. 0 means that there's no transport through the membranes. And what that root membrane looks like, so this is a cross section of a root in schematic version, is that for water to get in from the outside, from the bulk soil of that, how does the contaminant get into the xylem flow, where once it's in this xylem flow, there's no restriction to transport. There's no other membrane to move through. until it gets up to the leaf surface. There is absorption, of course, because this is an organic structure, but being fairly hydrophobic as far as the lignin in the tissue goes, many compounds, once they're in this flow, can move basically in and out of the screen, if you will, in a third dimension. And the question is, which compounds move in, and then what's the impact on the biosphere or food chain security for one issue? So then related to some work that was done by Lipinski that looked at, do drugs or pharmaceuticals that you take orally get into the body? So essentially it was a measure of permeability of intestinal membranes. So they screened a lot of compounds, and the rule of 5 from Lipinski's work is essentially if the compound has a KOW, log KOW less than 5, a molecular mass less than 500 Daltons, 5 less than 5 hydrogen bond donors or greater than 10, so it's not a perfect 5 here, less than 10 hydrogen bond acceptors, it is likely to move in through the membranes. So then this was cited by over 25,000 citations. So that kind of got my attention of this is some pretty important and good work. And then we said, well, does this permeability of intestinal membranes in mammals relate to the root permeability for different compounds? So again, Matt did the majority of this work, but it essentially binned the data for plant uptake studies and said just based on log KOW, again, we did see this sigmoid, or it's not sigmoid, excuse me, bell-shaped curve where around log KOW of 2 to 3, we see a majority of studies show that there is uptake. And then we looked at molecular mass, and here's Lipinski's cutoff at 500 Daltons. And we saw that most of the uptake studies predicted compounds were migrating into roots at about 2 to 300. So a little bit more restrictive, perhaps. And again, there's just not as many studies up around that 500 either. Then we look at hydrogen bond donors. The lower the hydrogen bond donor number for a molecule, the more likely it was shown to be taken up. Hydrogen bond acceptors the same. And then we also looked at polar surface area and rotatable bonds. Some of the studies after Lipinski's work brought into those two molecular properties. And again, they had cutoffs published, not by Lipinski, but others. And we can see that the plant roots do look a little more restrictive, so to say, for compound translocation, as do intestinal membranes for resistance for organic molecules to move in. So this is really interesting, and this was published in the first issue of Environmental Science Technology Letters, and then that publication was highlighted in Science. We're pretty pleased with that. So again, just kind of looking at that, and we can look at what those membranes are like. In our future work, we want to understand, can we predict, essentially based on what the membrane properties are of the root structure, is it similar to that intestinal transport? So to better understand some of this, Majid Bagheri, who's now a professor at Savannah State, pulled together a lot of this data that we had done in our lab, as well as the literature studies, and he was going to look at the root concentration factor, the transpiration stream concentration factor, and then the likelihood to get into fruit, where then we're a little bit more concerned about food security. And he did this through some artificial intelligence operations and looked at over 300 measurements of 150 compounds using a variety of plants and pulled all of this data together and screened it for quality and then put it into a neural network. So essentially the compound properties then for each one were either shown to be measured or just pulled from the EPA ChemSpider site. So we looked at, again, the same series of molecular properties. So again, putting it into an AI neural network, looking at all the different combinations of these and seeing what was actually out there, we put it in through the data. So all of the data then was pulled into some training data and then the test analysis for the neural network. So looking at this using AI methods, it was much easier to screen out what happened. And I'll show the data in just a second, but it came out that the organic uptake was dominated in order by log KOW. So while that is a restrictive one, that is the most restrictive and shows the properties the best. Molecular weight does matter, secondary, and is still highly significant. And the hydrogen bond donors were third, and then not as much. The rotatable bonds, and then the were predictors for not just molecular, intestinal membrane, but was also a better predictor for the blood-brain barrier. So some of the more recent work, again, stemming from Lipinski's work, really got down to the blood-brain barrier, not just intestinal membranes. And we are fortunate to be contacted, once we published that work, by some others. So we contributed to a feature article, Environmental Science Technology, on machine learning for environmental contaminant issues. So what else? Basically, when it gets down to it, could we utilize plants to screen for food security? And it would make sense because that's it. But could it also be a screening method for transport? So really, when it gets down to it, the bilipid layer in our intestines, which also has more complexity, is very similar to the lipid or bilipid layer that's in plant roots. So when we look at plant roots and understand what that transmissivity of a compound is, it actually does mimic what we might see in mammalian systems as far as transport through the process, through the body. So when we break this down, we can say with some confidence now that that plant root cell membrane does mimic the mammalian membranes and the blood-brain barrier. It actually seems a little bit closer as far as how restrictive most root structures are with the membranes to be similar to that plant uptake. So rethinking this of plants, we can look at plants as being sentinels for pollution and also used in remediation. So without question, when you see the plants changing the hydraulic gradient, they do change the subsurface. They can drop the groundwater and help aerate the soil, which increases aerobic biodegradation, and we've shown that repeatedly. But we also have it then as a way to pull contaminants with the water and the vapor phase to the root structures where they can either be taken up by the plant, biodegraded in the rhizosphere, or we can utilize some other interventions, like subsurface reactive addition or changing the redox to be more higher degradation rates for certain pollutants. But plants also give us air, they purify our water, they take care of the soil food area, and they provide provisions. So I'll shift gears here just for a couple minutes and say, while plants are doing that environmental remediation and environmental sensing for us, they also provide all of the other benefits that we see out of plants, such as ecology, and give us social values. So when we think about this holistically, we not only have an engineering solution for an environmental problem, we've actually got essentially what we need, all the things on the screen, for society. So we're bringing in societal benefits at the same time we're bringing in other ones. So to kind of pull this together, I'll just say we're looking at some new ideas here, bringing in plants as far as an engineering tool for subsurface investigation and remediation. And it's really that core of new ideas essentially can be taken into a new world. So our motto here is changing the world, and we think through a novel approach we can do that. So to wrap it up, essentially, when we're looking at natural systems overall, and they can have a lot of other benefits, not just on organic and inorganic contaminants, but we can also do regulation of stormwater. We can do green roofs. We've got a green roof research facility here. So combined, we can actually change quite a bit in our urban environment, as well as just a remediation. And all of these things are then benefits that we get along with that, in addition to our core thing. So I'll wrap it up. Here's just a shout out for Shel Silverstein in a book, and a quote from Buddha that kind of brings it all together of plants provide us with a lot of things. And within that, I think we bring another benefit to it as far as being an environmental sensor and an environmental tool. And I'll leave you with the beauty of plants as well. Not only do we see the benefits that we have around us, we pull plants into our home to give us comfort. We have artistic versions of plants around us to pull that in. So I would say thank you for your time.
Thank you. Thank you
And I'll
so much.
leave this.
Fascinating. So the plants can